absence of a chair, I'll introduce myself. I'm Jeanne Loring. I'm from the Scripps Research Institute in La Jolla, California. And when I first talked to Aubrey about giving a talk here, I thought I was going to give a talk more like George Church's on epigenetics and genomics of stem cells, but instead he asked me if I would talk about our experiences in trying to push forward a stem cell therapy for Parkinson's disease. So at the other end of the spectrum, I'm going to talk about negotiating the minefields of trying to develop and establish a cell therapy for Parkinson's disease. So just to give you the primer on stem cells, once again, there are two kinds. There are multipotent stem cells that come from the cells like the bone marrow or fat cells. These are mesenchymal stem cells or hematopoietic stem cells. And there are pluripotent stem cells, which can give rise to every cell type in the body. And if you have trouble remembering this, I think this is a good way to visualize the difference between these two cell types. Multipotent stem cells can make a couple of cell types, whereas the embryonic stem cells and pluripotent stem cells can give rise to every cell type in the body. And if you need a different kind of reminder, there's this one. Pluripotency is the ability to become anything when they grow up. Okay, so this is another way of illustrating pluripotency, and if you look in the upper left-hand corner, dopamine neurons are one of these leaves on a tree that can emerge from pluripotent stem cells. Dopamine neurons are the cell type that die during as Parkinson's disease develops. And pluripotent stem cells, also to remind you, can come from two sources. The original source was embryos that were discarded after about 5 days after in vitro fertilization. The inner cell mass of those cells can give rise to embryonic stem cells, which are pluripotent. And the other source are skin biopsies or other tissues from volunteers, which you can use to grow cells in vitro, like dermal fibroblasts, which you can then use reprogramming factors as developed by Shinya Yamanaka to make those cells into cells that are indistinguishable from embryonic stem cells. They have the same capabilities. So why do we think the stem cells will work for Parkinson's disease? Well, this is really a historical argument because we know that transplants of fetal tissue in Parkinson's disease patients were performed in the 1980s and 1990s. And occasionally, this strategy worked very well. The problem was that it didn't work every single time, and sometimes the results were pretty disastrous. So what we decided to do was to try to use these new stem cell technologies to sort of reinvent the fetal cell transplants that were done in the 80s, but in a much more controlled, quality-controlled way and reproducible, and presumably more successful. So I think you know that loss of dopamine screen neurons, the substantia nigra, is a major cause of the symptoms of Parkinson's disease. And if I could point, which I can't, you can see that the target of those cells is the striatum, which is the caudate and putamen in the brain. So the idea behind the fetal tissue transplants, beginning in the 1980s in Sweden, was to take tiny little bits of tissue from fetuses, 6 to 10-week-old fetuses, and take just the very small part that would be the precursors for dopamine neurons, put several of those pieces together and transplant them into the target of the substantia nigra neurons, the striatum. And that's illustrated in this figure in the center, and then on the right you can see what those little orange things are supposed to be, the cells that have been transplanted. Oh yeah, thank you. I can't believe you carry one around with you. Oh, well, I'm a speaker too, and I didn't have one. Okay, never mind. Okay, so I'll try to speak into the microphone. So as you can see here, these little orange things are the dopamine neurons that would transplant straight into the target, which makes a lot of sense in this case because we don't want to ask these cells to regrow as they did in embryonic development because it's a lot longer way to go than it was when they first connected. Okay, so in the US, there were actually two major clinical trials using fetal tissue. One of them was done by Kurt Fried in Colorado. These are NIH-funded, double-blind studies, which means some people in the study did not receive the tissue, they just received the surgery. And in Kurt's study, they had 40 Parkinson's disease patients. In the other study, which was run by Thomas Freeman, also sponsored by the NIH, they had 34 patients. The studies were similar but not identical. The idea was to discover whether the surgery itself, what the effects of the surgery itself would have, as opposed to the transplant of the tissue. So in both cases, there were some people who recovered really well. In fact, in this book called Healing the Brain, that is really, I really recommend it. It's a personal account of Kurt Fried's experience in developing Parkinson's disease trials back in the 90s. Some of the people had long-lasting relief and stopped taking drugs, but in about 15% in each case, there was severe dyskinesia, that's lack of control of movements. And that was the main problem with these therapies. This just shows the data from 2 patients that continue to improve for many, many years. It also points out there's a very strong placebo effect, and you'll see right after the transplantation, this is the reduction in a score, which this is improvement going down. On this line, this is the increase in release of L-dopa, or released by dopamine by the cells. So you can see they improve immediately, and then they continue to improve, this one for 16 years, this one here for 13 years. And there was very recently a study on some of the same tissues that had been done in the early Swedish trials, a closer look. And it was found that after, in a number of subjects, these 2 are shown, are 9 years and 14 years after the transplants, you can actually see healthy dopamine neurons from the transplants. So when they work really well. However, here's where when the minefields came into play. There are quality control issues, the big-time quality control issues. You have to use multiple fetuses. Didn't really know exactly how many, but if you've ever imagined trying to collect multiple fetuses and then collect tiny bits of each one of those fetuses, that's not a trivial activity, especially in the U.S., where abortion is not exactly considered to be a viable activity. So the other issue is the fetal tissue viability varies. And I've seen this myself because back in 1987, I worked at a company called HANA Biologics, and we were actually trying to reproduce some of this work. We were trying to grow these little tiny pieces of tissue and make them bigger so we could treat more patients. It turned out bigger was not better, and the cells de-differentiated, and they weren't useless. But it was an interesting idea. It had to be tried. So we found that the fetal tissue was of huge variability, and you really couldn't, in some cases, even discern where the brain was. So it's very difficult to get good tissue. And then there's the complex legal and ethical landscape, of course. And then the real killer was the dyskinesias. If you're treating somebody, you really don't want them to get something worse than what they had to start with. So that was that caused a lot of problems, and this pretty much shut down this line of research for many years. And interestingly, in the interim, just in 2010, it was discovered, or at least reported, that the dyskinesias were caused by the presence of serotonergic neurons in the transplants, which is not surprising. You're taking a piece of tissue which is going to differentiate into a number of different cell types. It's not surprising to have another cell type in there. This is good news for us because whenever we can impose quality control, we can look for the absence of certain cell types that we don't want to have in the preparations. Okay, so pluripotent stem cells can be differentiated in dopamine neurons. They can be produced in large quantities. They can be produced under quality controlled conditions, and you can use defined number of cells, defined cell types. We can use GMP to grow the cells, and If you use the cells from the patient, him or herself, then there are no problems with, you don't require immune suppression at all. So the strategy we came up with we started this project in 2012, so we've only been doing it for 2 years. Andreas Bratt-Leal is the principal scientist on this project. He came to me as an engineer and turned into a stem cell researcher of great skill. We decided to make iPS cells, induced pluripotent stem cells, from skin biopsies of 8 Parkinson's disease patients. These patients were from my colleague's clinic at the Scripps Health, and I'll talk a little bit more about that in a second. We chose them very carefully based on the earlier studies. We wanted patients that were really committed, really interested in the therapy, wanted to get involved in the therapy, and were very physically fit. That makes a huge difference. So we have been able to develop dopaminergic neurons, the precursors, the stage at which we want to transplant the cells in culture relatively reproducibly. There's some challenges between cell lines, but we've managed to solve most of those, timing issues mostly. And then we've also been able to make, as I'll show you in a moment, mature dopaminergic neurons in culture, so we can show that these cells are actually capable of becoming mature dopamine neurons after they've been transplanted. And we've tested and shown that these cells are effective in the rat model of Parkinson's disease, in a rat model of Parkinson's disease. These are the 8 patients right here, across the bottom. They're in the lab a lot, which is a new experience for me. We've created, as I will show you in a moment, a unique partnership between patients, patient advocates, scientists, and physicians. Okay, so this is the way that we make the cells from iPS cells. We start with the iPS cells. We are using a modification of a method developed by Lorenz Studer's lab. Works really well. It allows us to have some interim stages in which we can check the cells. So we first turn them into what are called floor plate cells. That takes 11 days in culture. And then we turn them into immature dopaminergic neurons, and that takes up to 25 days in culture. That's the stage at which we transplant the cells. We can carry them on farther for 60 days or more to make mature dopaminergic neurons in a culture dish. So we have quality control criteria at each stage. So obviously for iPS cells, we want them to show positivity for pluripotency-associated markers. We don't want them to be showing differentiation of or showing expression of other differentiated markers. There's a set of markers for the the floor plate stage. We do parallel cultures so that we can actually stain one culture and keep the other one. We stain the culture, and if they don't show up to be positive for these markers, or if they have a lot of contamination with other cells, we ditch the experiment right there. We don't carry it on. And then the markers for the stage at which we transplant the cells are these dopaminergic associated markers. But we want them also to be negative for things like serotonin. We don't want neural crest cells. And these 2 markers, for example, and GABAergic cells. These are immature neural progenitors, forebrain progenitors. These are neural crests, this is serotonin, these are GABA. These are things we don't want, so we want to be negative for those. And then we confirm that the cells are actually dopaminergic neurons by using patch clamp electrophysiology, which I'll show you in a moment. So what we're working on right now, because we really want to nail the release criteria for these cells. We want to know exactly what's going on in the cultures every single time. We've developed a transcriptome analysis for pluripotent cells, which is very popular. It's called pluritest. We also have epigenomic analysis of our pluripotent cells. So at the floor plate stage, we've gathered the data, but not quite enough yet for the transcriptome and epigenome, and we'll be doing that as our release criterion rather than immunocytochemistry. And then when the cells are ready for transplant, we're going to be adding data we're collecting right now on single cell analysis, because we want to get an idea about the heterogeneity of the cells at this stage. How many different cell types are there? And then since we can differentiate them into dopaminergic neurons, we're taking advantage of that by by grabbing single cells, and we want to identify new biomarkers for dopaminergic neurons. This just shows that these are our very first studies in rats. The red line and the greenish line are patient cell lines. The vehicle control is in blue. What you want to see here is a decrease in the rotations in this animal model. This is a very old and established animal model. And you can see that the cells do quite well in this assay. They've only done 2 cell lines so far, and we're moving forward with more. So these cells were actually frozen and thawed. We sent them, they were done by a contract research organization, which Charles River, which happens to be based in Finland. So this gave us an extra sort of variable. We had to freeze the cells, ship them to Finland, have them thaw them. We had to teach them how to do viability assays. And then, of course, they knew how to do so it was, I think, a really good way for us to test the sort of how far we could go without screwing up. So we want to use the patient's own cells, and this is controversial because this is going to make a much larger challenge for us when we talk to the FDA, and it also is much more of a challenge because we have to get the same results from every cell line. But there is evidence that a patient's own cells are likely to be better for treating Parkinson's disease, and this is the best data to date. This work was done by Jun Takahashi, who's doing a similar project in Japan, and he took autologous and allogeneic dopaminergic neurons, same stage we're using. He has a very similar method we do. And he found that if you transplanted the cells back into the same monkey you got them from, the cells survived very well. There was no immune response. However, if you transplant those into a different monkey, then you would have less survival and there would be an immune response. So it's not that it's impossible to use allogeneic cells, it's just that it's clearly better. So we're hoping that we can actually use the best possible method. So I told you this is how we can tell that these are dopaminergic neurons. We are lucky enough to have a colleague in the same building who has been studying the substantia nigra dopaminergic neurons in mice and in slice cultures. So he's patch clamped a lot of them. When he patch clamped our cells in culture, he was absolutely blown away because the pattern is exactly the same. So it looks like we have, this is a unique signature of a dopamine neuron from a human in its mature state as we would expect it to be in the brain. So these cells are capable of making fully mature neurons. So I told you we were planning to, now if George Church were here, he would tell me something which I'm gonna leave this with you guys because I don't know if any of you are gonna catch this or not. So when you do patch clamp, one of the cool things you can do, and if you look at this cell right here, you can see its cytoplasm is being sucked out of it. So we're collecting the cytoplasm from the cells that we've identified by electrophysiology, and we're doing a whole transcriptome analysis on that cytoplasm to see what that particular cell with that particular electrophysiological signature is making. And nobody's ever done that before, so we're expecting to find some new things that haven't been identified before. Okay, so here's where we are now. We've made iPS cells from 8 Parkinson's disease patients. We've made dopaminergic neurons so far. We've managed to sort of finagle the technology so that we can get reproducible cells from 4 different patients. We've transplanted 2 of them. Our QC methods in development right now are the genomic signatures, the single cell analysis. But it's very important that I've established collaborations with 3 other groups that are doing transplants of cellular therapy for Parkinson's disease. One is Lorenz Studer. He's using embryonic stem cell derivatives. Roger Barker in UK and the European Union is repeating the fetal tissue studies, but with much higher control over the quality of the tissue. And then Jun Takahashi is doing a similar project to ours, little variant, on iPS cells in Japan. And all 3 of these, Roger's already sent me the tissue. I'm hounding Lorenz right now for his cells. And Jun Takahashi has promised that he'll provide with me the cells as soon as he gets them exactly perfect. Okay, so here are our current challenges. Fundraising is a big challenge. We plan to have a pre-IND with the FDA this year. There's still some of the year left, so I think that's feasible. We have to do more fundraising and then a pre-IND, which we'd like to do in 2 years. That depends on whether the fundraising worked, and then we have to do more of that. We're then going to get feedback from the FDA and spend some time satisfying their recommendations. And they were hoping for an IND in 2017. Now that's feasible if we meet certain funding challenges. And then we do more fundraising. And ultimately clinical testing. So this is an unusual project in that it is funded entirely right now with private funding from a foundation that was founded just for this, this purpose. It's a collaboration scientifically with myself and Melissa Hauser, who's the director of the Movement Disorders Clinic at Scripps Health. These are her patients. This woman who's standing in the front of the picture is Sherry Gould, and she's the spark plug of this. We call this Summit for Stem Cell, and you'll understand why in just a second. It's a true partnership of patients, scientists, physicians, and donors. The scientists come in and visit the lab. We give lab tours every week to potential donors, scientists, people who just show up off the street, because we really want to educate the community about this very, very straightforward and simple stem cell strategy. So the funding we've done so far is from small donations. This is our giant check, which is on the left. We got $200,000 from a fundraising effort from the Parkinson's Foundation. And then on the right, just a few weeks ago, we got an amazing almost $1 million at one dinner in Solana Beach for this project. All this money is going into a fund, which we're keeping, because we're going to use it for matching funds for a serum grant that we're applying for. So we continue to just sort of step along. So I wanted to close with this because I wanted you to get an idea of what these patients are doing in order to. Now you probably won't hear the sound because I haven't turned it on, but you won't hear it anyway. You can hear it. Okay. This is a trip, this is the second of the mountain climbing expeditions that the patients and their families and their advocates took. The first one was to climb to Mount Kilimanjaro. The second one was to climb to Everest Base Camp. That's this one. These are people with Parkinson's disease, many of them. And you can actually tell in some cases that they have Parkinson's disease. Okay, so that gives you an example of what an adventure this is, also for me. Unfortunately, I had to work because so I couldn't go on the Everest trip with them. I'm hoping they go somewhere else really interesting so I can actually go along next time. But they are in really good shape, and it goes along with the hope that they have and the understanding that they have of this therapy. Any one of those patients could have stood up here and given the same talk that I can give. So that gives you an example of how you can push forward a therapy with the help of a lot of friends and the patient advocates, the people who actually will be treated by this therapy. Thank you.